the cell structure and functions, we have studied various types of organelles and their functions. After studying the different organelles and their functions, now we shall study about the cell cycle and cell division. Now just to recall what we have studied related to cell previously, cell is the unit of life, and the various parts of the cell like cell wall, cell membrane, mitochondria, Golgi complex, endoplasmic reticulum, ribosomes, lysosomes, then mitochondria, plastids, vacuoles, cytoskeleton, cilia, flagella, and centrosomes. So all these are the organelles which are present in a typical cell. Now these organelles, they are having their own functions. So there is a kind of coordination of work, or there is coordination between the organelles to make a stability in the function of the cell. So cell is the basic unit of life, and how this cell divides, what are the stages, types of cell division, we shall study in this part. Now cell cycle and cell division. Now during cell division, there is a kind of various kinds of events, sequence. So this sequence of events by which the cell divides. So actually there is duplication of the genome in its cell, and also the other constituents of the cell, and then the cell divides into two daughter cells. So these events constitute the cell cycle. So the cell cycle is the sequence of the events which takes place in the duplication of the genome. Now genome means actually the amount of DNA. Actually in the DNA there are genes which we call when we go into more detail about DNA. So DNA undergoes duplication. So there is duplication of the genome, and also the organelles, they increase in number, and these events which takes place to divide the cell, and two daughter cells are formed. So then this is called as cell cycle. Now the cell cycle is the one which involves or which constitutes all such events which increase the number of the organelles and all that. Now this events which takes place, all these takes place in a coordination within the cell. So already I have mentioned that the organelles present in the cell, they perform their own function, and this function which takes place in the cell, so there is a kind of coordination between the organelles, and thus it maintains a kind of stability in the functioning of the cell. Now however, all these events which takes place, so all the functions which are taking place in the cell, they are under the genetic control. So if you remember the nucleus when we have studied in the previous part, so the nucleus is the controlling center of the cell activities we had mentioned. So the function of the nucleus is, it is the controlling center of the cell activities. So that means all because of the DNA. So this nucleus has been the center, controlling center, because due to the presence of the DNA in the chromatin or chromosomes. That is why the DNA is having genes, so that genetic control is done by the DNA. Whatever functions which are to be undergone or to be taking place in the cell, it is all due to the DNA. So this cell cycle takes place in living organisms. So and what are the steps or the events in this cell cycle we shall study. So the cell cycle is divided into two basic phases, that is the interphase and M phase, or which is also called as mitotic phase. So M phase, it is the mitotic phase. Now interphase and M phase, mitotic phase, includes the cell cycle. Now this cell cycle actually it is the events which occur in the division. So when we come to the cell division, so there are two types of cell division, mitosis and meiosis. Basically we know that there are two types of cell division, mitosis and meiosis. So we will be going to study about mitosis and meiosis. So but actually when we go further into detail, there is also one more division. There are three divisions, types of cell divisions: amitosis, mitosis, meiosis and amitosis. But usually amitosis takes place in some unicellular organisms, some lower organisms. So that is why whenever we speak of types of cell division, we usually mention only two, because mitosis and the meiosis are the more commonly occurring types of cell division in all living organisms. That is why there are two types of cell divisions, mitosis and meiosis. So the now cell cycle is divided into interphase and M phase, mitotic phase. Now whenever a cell starts dividing, suppose this is a cell. So whether it is a plant cell or animal cell, so the cell divides, and whenever it starts to divide, so it prepares for cell division. So in other words, preparatory phase, what we call it as. So that preparatory phase is called as interphase. So it is common that whenever we want to do any function, we prepare to do that function. We prepare ourselves. So there are some basic requirements for the preparation of a particular function. Similarly, whenever a cell is to be divided, suppose this is a cell. So if this cell need to divide, or there is a necessity of cell division, so then that cell will prepare for division. Now this cell division, when it is called as a preparation phase, preparatory phase or interphase. So what are the preparations which are to be done? So this interphase has again three steps. Main three steps are there in the interphase. So those three steps are: one is G1 phase or gap 1 phase, synthetic phase, synthesis phase or S phase, gap 2 phase or G2 phase. So these are the three steps of interphase where the cell prepares. So any function which is to be done, preparation is necessary, even for us also. Whenever we want to study or whenever we want to go through anything for the exam, so we prepare. So that preparation phase is nothing but the interphase for the exam or the preparation phase. So here in the interphase, there are three steps occurring in the cell: G1 phase, gap 1 phase, synthesis phase or S phase, gap 2 phase and G2 phase. Now these three phases which we cannot see morphologically in the cell, but however they are all molecular changes going on in the cell. Now for example, in the gap 1 phase, what happens? There is a synthesis of RNA and proteins. So RNA and proteins are synthesized in this stage, and the cell which is normal, it somewhat grows to its maximum size. So here the growth of the cell to its maximum size means it does not increase remarkably. There is no increase in the size of the cell to a great extent. To a certain extent, or even to a small extent, we can say the cell will grow and it will become metabolically active. So the cell becomes metabolically active, and thus the cell will grow to its maximum size. That is the first phase where the G1 phase takes place. And also during this phase, proteins and RNA are synthesized. So this step, so all these three steps are actually very short, but they are specific to certain functions. So proteins and RNA are synthesized, the cell becomes metabolically active and grows to its maximum size. That is what actually occurring in this phase, gap 1 phase it is called as. It is also called as G1 phase. And in the second phase, that is a synthesis or S phase. So there the synthesis phase or the S phase, so the word itself suggests there is something synthesis of material. It is nothing but the synthesis of the DNA. So the DNA will get prepared, or in other words, what happens? The existing DNA will increase and it will become double its quantity. So in other words, it is also called as the DNA replication. So the DNA quantity increases and thus the DNA synthesis takes place. Purely there is DNA synthesis. So this is the only function in this. And also another important function is the centrioles, they undergo duplication. The centrioles become double in number, and that is also another important event that takes place during the S phase. So in the cytoplasm, the DNA will increase and the centrioles become double, or they become increased in number. That is synthesis phase. There is no RNA synthesis, no protein synthesis. There is only DNA. That is synthetic phase or synthesis phase, second phase. Now in the G2 phase or gap 2 phase, what happens? So there again some repetition of the events takes place here. The synthesis of RNA and proteins had occurred in the G1 phase, again it will continue in the G2 phase. Now in the G1 phase, proteins and RNA were synthesized, and it stopped for a short moment, and then DNA synthesis will occur in the next step, S phase. Now after S phase, the DNA synthesis will stop. So when the DNA becomes double its quantity or increases in quantity by replication, so there the DNA synthesis will stop. Now after the DNA synthesis stop, there the S phase completes. Now after that, RNA synthesis and protein synthesis will continue, which had occurred in the G1 phase. Now this is the G2 phase. Now that is why we call it as a G1 and G2. So G1 means it is the first phase where RNA and proteins have been synthesized. It is one step. In the G2, that means the second step, where again proteins and RNA will synthesize. In between, we are having the DNA synthesis, which we call it as the synthesis phase. So that means the formation of proteins, the formation of RNA is very important. So the proteins are necessary for the formation of the spindle fibers during cell division. That is why the protein synthesis and RNA synthesis occurs in two steps. So here also protein and RNA, here also protein and RNA, DNA only in the S phase. There is no DNA synthesis here. The DNA synthesis will not continue in the G2 phase. It will stop. When the DNA synthesis stops, there the S phase stops. And next the RNA and proteins will continue. That is what occurs in the this G2 phase or the gap 2 phase. Now after this three phases of the means, now the cell is prepared. It is ready for division. Now in some cases the cell continues to divide after G2 phase, but sometimes the division is not necessary at the present moment. The cell is prepared, the cell has prepared for division, but soon after preparation it may not start dividing. So that is why when it is necessary, when the division is needed, then the division will start. That means after G2 phase there is a short gap where the cell will not divide. So that is called as the Z0 phase, or it is also called as quiescent phase. It is called as quiescent phase. Now quiescent phase means it is the stage where the cell remain metabolically active, because in the G1 phase the cell is metabolically active. So that activity will remain as it is. There is no decrease in the activity. It will remain as it is. And also the during the G0 phase, what happens after gap 2 phase? The cell is ready for division, so it will remain metabolically active, and this cell will not undergo any further change in the organelles or in the synthesis of any DNA or RNA or proteins or like that. And only when the need of cell division is there, then until that call occurs, then it will not divide. This call actually is done by the DNA, genetic control. So unless it is called upon to do so, that means to do the further cell division, so depending upon the requirement that occurs, if there is need, if there is requirement for the cell division, then only there is genetic involvement for cell division, and thus the cell will divide. But however, this G0 phase is somewhat a resting phase, hence it is called as quiescent phase. So these three are the activities. This is a somewhat a resting phase, G0 or quiescent phase it is also called as. Then after this we come to the next stage, that is the M phase or mitotic phase. So the M phase or mitotic phase is actually the division of the cell. So the cell divides and form the daughter cells. So in the mitotic phase, we are having two, broadly it is divided into two phases, like karyokinesis and cytokinesis. Karyokinesis is one step under mitotic phase. Under interphase 3 main, G0 phase may occur or may not. Interphase means G1 phase, S phase, G2 phase. Now after that there is actually M phase, but sometimes the cell will remain metabolically active, and only if there is a need or a requirement of the division, then it will divide. So until then there is the quiescent phase or the G0 phase. Then karyokinesis, then the M phase is divided into karyokinesis and cytokinesis. So these are the two steps of the cell division: karyokinesis and cytokinesis. Now in karyokinesis again there are 4 steps. So under first one, the one is prophase. Under karyokinesis, prophase, metaphase, anaphase, telophase. So basically we have come across in the lower classes the cell division. So these terms are not new to us. So the karyokinesis means actually the division of the nucleus. So the cell, after preparation, the nucleus will first divide. So this nucleus of the division to divide to form 2 nuclei is called karyokinesis. After the division of the nucleus, the cytoplasm will divide, that is cytokinesis. So the word itself suggests kinesis means break, karyo means nucleus, which is a Greek term. So this karyo means nucleus, monokaryotic, dikaryotic, usually we call it as. When a cell is having single nucleus, monokaryotic, 2 nuclei, dikaryotic, like that. So here the nucleus divides by 4 steps, by prophase, metaphase, anaphase, telophase. Now in the prophase, what actually takes place, we shall see. Now in the prophase, now suppose this is the cell, a prophase cell. Now this prophase, in this, the nuclear membrane, the changes which takes place. The nuclear membrane, they break into small segments like this. So it will break into small vesicles like. So already we have studied about the nucleus, which is called as the nuclear envelope. So the nuclear membrane will break into, or it will break down into small membranous vesicles. So these are membranous parts of the nuclear membrane which break down and remain in the cell. This is one important part. Then the chromatin present in the cell. Now this chromatin, suppose this is the chromatin, they start condensing to form chromosomes like this. So already in the nucleus, after the nucleus we have studied about chromosomes, the types of chromosomes and all that. So the chromatin starts condensing during prophase. So as it starts condensing, the chromatin becomes thick and condensed to form a rod-like structure called as a chromosome. So chromosomes are formed in the prophase. Suppose these are the chromosomes. So this is one chromosome and this is another chromosome. So I have shown here 4 chromosomes in this cell. So the number of chromosomes is constant for a species. In human being there are 46 chromosomes, and like that in other organisms the number goes on differing. So the chromatin undergoes condensation. The nucleolus disappears, and the nucleolus, which is a dark spherical body in the nucleus, and that disappears, and the nuclear membrane breaks down into small membranous vesicles, and we can see the discontinuous nuclear membrane in the cell. The chromatin becomes chromosomes, and here when the chromatin becomes chromosomes, the centriole, which had duplicated in the S phase, so this centriole will get arranged in the polar end of the cell. Now suppose here this is a centriole, this is a centriole. So this centriole, this is a centriole. So these are the chromosomes. So these are the chromosomes, and this is the centriole, which become arranged at the either ends of the cell, which we call it as the polar ends. Now when the centriole become arranged, they give rise to small fibers on its outer surface like this. So these small fibers, they radiate from the centriole, and they appear somewhat a star-like structure. So this structure is called as aster. So this is referred as aster. So these are the events which takes place in the prophase. So 3 main events we can mention in the prophase. The chromatin condenses to form chromosomes, but however the condensation is not complete. There is maximum condensation, but the condensation is still going on, but we can see the chromosomes in the prophase. This is one important change. Then the nuclear membrane breaks down at different intervals into small vesicle-like structures called membranous vesicles. So there is discontinuous nuclear membrane in the cell, and also the nucleolus and other structures, they disappear. So this takes place in the cell. Then the centrioles, they arrange at the polar ends of the cell. Now these centrioles, which get arranged at the polar ends, so they produce fine fiber-like structures. So these fibers, which are called as the microtubules, they radiate from the centriole. Now actually here these microtubules, they are present in the centrioles. So they are arranged in triplets. So those microtubules are proteinaceous, that is taking place. Then one more we can mention in the prophase, the viscosity of the cytoplasm or the cell increases. So this viscosity of the cytoplasm increases. That means the liquid cytoplasm becomes somewhat viscous in nature, and thus the viscosity increases in that cell. So 4 events which we can mention in the prophase. So just to repeat, chromatin becomes chromosomes, one important point. Nuclear membrane breaks down to form membranous vesicles so that the nuclear membrane is discontinuous. Then third one, centrioles arrange themselves at the opposite poles of the cell and they produce fine microtubules which are radiating from each centriole. So which we commonly call this structure as the aster. Then the viscosity of the cytoplasm increases. So these are the 4 changes which we can mention in the prophase stage. Then in the second stage, that is metaphase under karyokinesis. So in the metaphase, the condensation is complete. The chromosomes completely gets condensed. The nuclear membrane gets completely disappears. It completely gets disintegrated, in other words. So there is complete disappearance of the nuclear membrane. So there will be no nuclear membrane, and the chromosomes, they arrange themselves in the equatorial plane of the cell. This is the chromosomes, how they appear, and there is no nuclear membrane, and the microtubules, they radiate, they extend in all directions of the cell like this. So these are called as spindle fibers. So these are called as spindle fibers or mitotic apparatus. Spindle fibers or mitotic apparatus. So the chromosomes become arranged in the equatorial plane of the cell. So this is the equatorial plane, that is in the central region. The chromosomes are completely condensed. Suppose this is a chromosome. Now in the metaphase, what happens? You can see there is still more condensation of chromosomes like this. So you can come to see the difference here. This was the chromatin before prophase, and the chromatin becomes chromosome. It starts condensation. So of course chromosomes are seen, but the condensation is yet still continued after prophase. So during metaphase, the condensation is complete. So they are completely condensed. So here this was the chromosome. So now it is still more condensed and very clear. So chromosomes are very clear and distinct during the metaphase of the stage, cell division stage. So chromosomes, whenever we want to study, and even after metaphase, during anaphase, the chromosomes are not clear. Only maximum condensation or complete condensation is seen during metaphase. So whenever research work or anything we want to study about chromosomes, about its structure or any other characteristics about chromosomes, so chromosomes are selected or they are taken only from the metaphase stage. So during metaphase, the chromosomes are very clear. That is during metaphase stage, where the chromosomes are maximum condensed. And not only maximum condensed, they are arranged in the equatorial plane of the cell. Now why they become arranged in the equatorial plane of the cell? Now the question. Now suppose this is the chromosome. Now this chromosome, which is having a centromere. So this is the chromatid and this is the chromatid. So this is the chromatid. This is the primary construction or centromere. Now these spindle fibers, which is also called as the mitotic apparatus, they spread in all directions in the cytoplasm of the cell. So when they spread in all directions, some of the fibers which are in different directions, some of the fibers they attach to the, suppose these are the fibers which are spreading. So some of the fibers attach to the centromere. So they attach to the centromere. Other fibers, they are just in contact with the chromatid, and other fibers are still freely present in the cytoplasm like this. So certain fibers are in contact with the chromatid, certain fibers with the centromere, certain fibers freely present in the cytoplasm. Now those which are in contact with the centromere, they become attached to the centromere. So this fiber gets attached to this centromere. So from this direction and from this direction. So fibers coming from this end, they attach to the centromere, and also from this end they attach to the centromere. So if you remember, in the centromere there are two cap-like structures, kinetochores. So to the kinetochores the attachment is seen in the centromere. And after attachment, these fibers, spindle fibers, they start contracting. So this fiber also will contract, and this fiber also will start contracting. Now as a result of the contraction of the fibers, so there is pulling of the chromosomes to the either ends of the cell. So that means when we pull an object at both ends, suppose we are pulled from there to this direction and to this direction. So that means we will be moving either this side or this side. So during this movement, at one stage the chromosomes become arranged in the equatorial plane of the cell. That is metaphase. So chromosomes which were like this, the fibers stretch and this membrane is disappeared. So these fibers, they attach to the centromeres from both ends. So the fibers start pulling the chromosomes. So when the chromosomes are dragged either this side or that side, at one moment, at a particular moment or a stage, we can see the chromosomes become to be appearing in the equatorial plane of the cell. So that is that stage is called as metaphase. So this is metaphase. So presence of chromosomes in the equatorial plane, the complete formation of the spindle fibers. The spindle fibers, they attach to the centromere and pull the centromere due to the contraction of the spindle fibers, and that is during metaphase. So this is in the metaphase. So chromosomes are very clear at this stage, and we can count the number of the chromosomes. So that is this metaphase stage. In the third stage, that is anaphase. So during anaphase, what happens? So the stage in this, so these are the centromeres which have pulled the chromosomes like this in all directions. Now when the chromosomes are pulled here, these chromosomes, they are split longitudinally like this. So whenever the chromosome is pulled either this way or this way, so the movement of the chromosome takes place there, and they appear to be present in the equatorial plane. And later what happens? The chromosome gets split longitudinally, lengthwise, and this chromosome divides into two parts. So just by splitting a stick or anything into two parts longitudinally, suppose this is the chromosome, if we divide this longitudinally, it becomes two parts, two halves, and so the chromosomes become double in number. So each chromosome divides into two, so there will be 8 chromosomes. So this half is pulled by this fiber, this half is pulled by this fiber. So during the pulling of this fiber, the chromosomes appear to be bent like this. So this is the other half of the chromosome. Now actually these two were single chromosome. So when this was split and this is pulled, so the tips will bend like this. This is how the chromosomes look like. So they appear to be V-shaped or somewhat J-shaped. Sometimes they appear to be J-shaped. So J-shaped means like this, or V-shaped. This is how the chromosomes look like. So this is the chromosomes which are split. So the fibers are there which are attached to the chromosome, and they are pulling the chromosomes to the opposite ends. So this is the equatorial plane. The chromosomes now separate due to the splitting of the chromatids. So the chromatids, each chromatid will split, and also the centromere also will split. And after the splitting of the chromosome, they will become double in number. So out of each chromosome, one half will move towards one side, the other half towards the opposite side, opposite pole. So this and this was one chromosome. It has a split and it is moving towards the opposite directions. And in addition to this, there are certain fibers which are present in between the chromosomes which have been split. So these chromosomes which have been split, and here the certain fibers are left freely in the equatorial plane. They are called as the interzonal fibers. So these are called as the interzonal fibers. The fibers which are not attached to any chromosome, they are freely present in the equatorial plane, interzonal fibers. So other chromosomes, other fibers are there, they are attached to the chromosomes. So these chromosomes which are in two equal groups, they move towards the opposite ends, and this stage is called as anaphase. So in anaphase what happens, the chromosomes which were in the equatorial plane, they split into two, so that the existing number of chromosomes become double, and each chromosome segment, they move away from the equatorial plane to the opposite directions, and when they are moving, which they are pulled by the spindle fibers, they appear to be V-shaped or somewhat J-shaped, capital V or capital J alphabet shaped, so that appearance of the V-shaped chromosome or J-shaped chromosome, which are moving away from the equatorial plane towards the polar regions, so that chromosomes which are on the way, which are moving towards the polar ends, that is called as the anaphase stage. So in the anaphase, there are two groups of chromosomes, equal number of chromosomes, which are V or J shaped and moving towards the polar ends, and in between we find the interzonal fibers. So in the last stage, we are having the telophase stage, and where the chromosomes reach the polar ends. So the chromosomes reach the polar ends here, and these chromosomes, when they reach the polar ends, they become chromatin like this. So this chromatin, the chromosomes are not seen in the telophase. So after metaphase, the chromosomes will start converting into chromatin. So during telophase what happens, the chromatin, the chromosomes decondense. Chromatin condense into chromosomes. Now the chromosomes decondense, means they stretch as usual. So after reaching the respective poles, So when this group reaches this pole, when this group reaches this pole, the chromosomes will decondense and they will lose their structure of the chromosome. They lose their individuality and they become chromatin. So this chromatin material remains at the two opposite ends of the cell, and then a nuclear membrane is formed around each mass of chromatin. Now this is actually chromatin. So this is the chromatin, and two nuclei are formed. So here these are the two nuclei which are formed in this. That is telophase. So after anaphase, the chromosomes which are in two groups, they reach the opposite poles. The chromosomes decondense to form chromatin. They lose their normal structure, they lose their individual nature, and then they become chromatin. So after become chromatin, the nuclear membrane reappears, means it forms around each chromatin clusters, or sometimes the chromosome clusters they are also called as, and then the nucleolus in the nucleus appears there. The nucleus which had disappeared during the starting of the prophase, it reappears, and also the endoplasmic reticulum and the Golgi complex, they are also formed in this cell. That is telophase. So these are the four stages of karyokinesis. So the nucleus divide, the division of the nucleus is called as karyokinesis. So this karyokinesis is division of the nucleus. Now after karyokinesis, the cytokinesis takes place. So the nucleus which was single, it divides into two. See, the chromosome which has been divided into two, there is no any decrease in the size of the chromosomes. So that is very important, we should note. So here there is chromosome, here there is chromosome, here the chromosome has split. Even after splitting the chromosome will not decrease in size, because the amount of DNA has been increased during G2 phase, during S phase. During S phase, we had mentioned that there is duplication of the, or there is a DNA synthesis. So the DNA increases in quantity, centriole of course also duplication takes place. So that existing DNA which becomes a double, so in each chromosome we are having a double amount of DNA than previous, and that is why when the chromosome gets divided, again the amount of DNA remains constant. That is why there is no decrease in the size of the chromosome. So the chromatin also remains as it is. Now in the next step, the cytoplasm will divide, that is cytokinesis. Now in animals and plants, the cytokinesis is somewhat different. So in plants, the cytokinesis, now suppose this is the cell. So these are the two nuclei. The same thing I have drawn. Now cytokinesis in animal cells and plant cells is different. The division of the cytoplasm means when the cytoplasm divides, two cells are formed. So in animal and plant cells it is different. In animal cells, first we shall take. So in animal cells, what happens? The cell membrane will grow inwards at the two opposite ends like this. So this depression which occurs, it is called as groove, or it is called as constriction. So the cell membrane will grow inwards, it will form a depression here, and this groove will grow inward inside. So when it grows inward like this, suppose this is the cell, so the groove grow the groove increases or it deepens into the cytoplasm. So this groove or constriction, it will deepen into the cell cytoplasm. So both will move in the opposite direction, and then ultimately the two grooves, they meet each other, and thus the cytoplasm divides into two, and ultimately these two join and form two separate cells. So this is in case of animal cells. So in animal cells, cytokinesis takes place by the appearance of the groove or constriction at the opposite ends of the cell. However, cytokinesis means it is the division of the cytoplasm, so that the cytoplasm divides into two parts or two halves, or the cell divides. So this in the case of animal cell, but in the case of plant cell, this is different. Here what happens, the cell, suppose this is the cell, these are the two nuclei after telophase. Here there is formation of the cell wall. So this cell wall formation, it is due to the deposition of various chemicals, and it differs from a type of the cell. Sometimes it may be cellulose, it may be hemicellulose, or it may be lignin, or it may be pectin, or it may be suberin, or like that. So depending upon the material, this cell wall is deposited. Now the cell wall is actually deposited in the center here, and this cell wall will increase in both directions, and completely so this is first how it is formed. Then it will increase at both ends. These are the, so this cell, this is called as cell plate, it is called as. And in addition to this, the Golgi complex, the vesicles of the Golgi, they get deposited in this region, and after the deposition of this cell wall, here there is the formation of the middle lamella. So the middle lamella, or the structure in between the two cells. So after the formation of the cell wall, the Golgi vesicles, they also aggregate, so that Golgi vesicles, they increase and they get deposited in the center of the cell, and they are collected at the equatorial plane and get deposited with various chemicals. So these Golgi vesicles, they fuse and get deposited with certain chemicals, and it forms the middle lamella. So on this middle lamella, so the cellulose gets deposited, which forms the primary wall, or other words, the pectin may get deposited, so that forms the primary wall. So like that, a primary wall gets formed on the middle lamella on either sides of the cell, daughter cells. So here it may be cellulose or pectin, so two daughter cells are formed. So in plant cell, the cell wall is formed, there is no constriction. Here constriction cannot be formed due to the presence of the cell wall, and the cell plate, or sometimes it is also called as a phragmoplast, so that appears as a thick plate in the center, which extends in both directions and divides the cytoplasm into two parts. So that is about the cytokinesis in animal cells and plant cells. So this is about the M phase. So in the M phase, we are having the four stages, means two main stages, karyokinesis and cytokinesis, prophase, metaphase, anaphase, telophase. It involves the division of the nucleus, which we call it as karyokinesis. After this karyokinesis, after telophase, the cytokinesis takes place, which is the division of the cytoplasm. So in animal cells and plant cells, this is somewhat different, and that is because of the absence of cell wall and presence of the cell wall. And lastly, we come to what is the significance of mitosis. What is the significance of mitosis? Now, about cytokinesis, one more point. Now, cytokinesis means the division of the cytoplasm. Now, if this telophase does not occur, cytokinesis, imagine. If there is no cytokinesis, this remains as a single cell with 2 nuclei, isn't it? Suppose if this occurs, then only there will be formation of 2 daughter cells. Again, if cell division takes place in this, so this divides into 2 nuclei, this into 2 nuclei, only the nucleus divides, not the cell. So if in some cases there will be no cytokinesis at all. So in the endosperm of coconut, there is such kind of means absence of cytokinesis. Means cytokinesis is not seen. Cytokinesis is completely absent in the endosperm of coconut. So in the coconut milk, the coconut water, what we say, ಎಳ್ನೀರು ಅಂತ ನಾವು ಕರಿತೀವಿ. So that coconut water is nothing but it is a collection of nuclei which are freely present. So because of the freely occurring nuclei, that is liquid state. But when the same nuclei become nucleated, means when there is cytokinesis, so there are cells formed there, many cells are formed, so that becomes solid. So that is what actually happens in the coconut. So this is about the cell division. Now coming to the significance of the mitosis. So the main thing is, in case of unicellular cells, unicellular organisms, mitosis is the method of reproduction. In case of amoeba or any other unicellular organism, there the nucleus divides. So whenever amoeba is to reproduce, it simply divides into 2 cells. That is nothing but mitosis. So here mitosis is the means it helps to reproduce and increase in number. So in unicellular organisms, that is the case. But in multicellular organisms like us, mitosis is necessary for growth and development. See, from childhood we grow to up to certain age. So that means growth. It is the increase in size of the organisms. So when we were child, we were small, and as we years go on, so we increase, or our height will increase, our body size will increase. So that means nothing but it is growth. So this growth and the development of the organs in our body, it is because of mitosis. So it is because of the increase in the number of the cells. So the organs in our body which were small, they have enlarged because of the increase in the number of the cells, and thus mitosis helps in growth and development of multicellular organisms. And then another important function is, now after the cell division, mitosis here, suppose this was the cell and it has become 2 cells. So here when we see this, we believe that the cells become small. Actually these cells will not remain as they are. They increase in size and attain the size of the parent. So these 2 cells will enlarge and become the size of the parent. So that means there is increase in the cytoplasm, organelles and all that. So it maintains the cytoplasm and the cell surface area. So when this was there, the surface area will be reduced. So this was the size of the parent cell. So again this will increase, and there is maintenance of the proper surface area and the volume ratio of the cell. So the volume of the cell and the surface area, this ratio is maintained by mitosis. So the mitosis means it is not simply division of the cell. After division, the daughter cells again attain, they reach, they grow to the size of the parent cell, and thus the surface area and the volume ratio is maintained by the help of the mitosis, the cell size. Then this mitosis also helps in the cell repair. That means whenever a particular part of our skin is lost, if the skin is lost, here new skin will grow by cell division. So if there is an injury or if there is a wound or like that, so those lost cells are means occupied. So there the existing skin cells, they divide and thus help in the this cell repair by replacing the old and the worn out cells with the new cells. So for example, in skin and also in any part of the alimentary canal, we can see such kind of cell repair. And even in wounds, whenever there is a wound, so the existing cells are lost by the disease. So there is the healing of the wound, we call it as. So this healing of the wound also is by mitosis. So in injury, there it is lost, but here this loss is due to damage of the cells by this disease. So this regeneration or the healing of the wound is done by the mitosis. And also this mitosis helps in to maintain a kind of genetic stability which is brought by equal distribution of the chromosomes. See here, when there are 4 chromosomes, so the 4 will split and 4 will remain in each nucleus. So there is no unequal distribution. So mitosis also brings about equal distribution of the chromosomes, equal distribution of the DNA. So what amount of DNA is here, the same amount of DNA is present here. So what amount of DNA is present in this nucleus, the same amount of DNA is present there. So that means this is called as genetic stability. There will be no any variation in the DNA. So there is uniformity. So this is maintained, this genetic stability is maintained by the equal distribution of the chromosomes during this cell division. So by mistake there is no any such disorder in the distribution. Suppose if there are 8 chromosomes, 5 on this side, 3 on this side, so that does not occur. So mitosis maintains genetic stability in case of this that is the cells where the daughter cells which are formed, they are both are same. There is no any that means unequal distribution of the chromosomes or the chromatin or the DNA, and even in the cell organelles also there is no such kind of unequal distribution, and thus the mitosis has various kinds of significance of mitosis. So this is about the mitosis, the process of mitosis, and interphase, M phase, and also the significance of mitosis. So mitosis is compulsorily present in all eukaryotes, because in prokaryotes usually mitosis is rare, because in prokaryotes there is no formation of spindle fibers. Of course there is a cell division. So whenever bacteria divide into 2 cells, it is, we call it as a simple cell division. We usually refer it as binary fission but not mitosis cell division. So but mitosis means it maintains a genetic stability, equal distribution of chromosomes. There is the maintenance of the cell size So the cell size is maintained, or there is also maintenance of the surface area and the volume ratio of the two cells. So that is the significance of mitosis. But in case of prokaryotes, we do not see such kind of mitosis because of the absence of spindle fibers.